Hello, champions. Let me see if it is loaded, not loaded. Yes, it's on. Children, a very good afternoon to all of you who have come over here. And also, just let's wait for 1 minute. Works? Hi Dhriti, hi Aarti. I see lots of children coming up there. Hello. Good afternoon. I'm properly audible, right? We'll just wait for a minute and then we'll start off. We'll just wait for all the children who want to join. I hope you all are doing great. You all are happy, you are healthy, and you are preparing really well for your examination, that is KCET. Hi Kiki, very good afternoon. So children, if there is someone out there, your friends who need help in biology, please send the video link to them so that they can also come and watch it. Hi Shamna. How many questions coming from practical? Probably, I think 2 or 3 could be coming. That's the maximum. This is the first time, so let's be hopeful that not much would be coming. Hi Muhammad Khizar. Hello Narasimha. Bhargavi. Okay, I see lots of children coming up. Shall we start? Okay, and children, all of you are doing good? You're happy, right? Give me a thumbs up if you're all happy. If you're all happy, not worried about anything. Okay, we are going to ace the examination. Yes. Hi Shivakumar. Okay, chalo, let's start. Let's start off with the first question. Hi Alan. Okay. So over here, I'll tell you what is going to be there in this particular video. So I have targeted certain experiments of your practicals, biology practicals, and on the basis of that, there will be few easy questions. Okay, it will be easy. I'm pretty sure it will be easy for you guys. Now, after that, I would be solving certain previous year questions of sexual reproduction in flowering plants and also human reproduction. Total, there will be 35 questions. There is 1 extra question. That's all. Okay. Apart from that, all is sorted. So let's begin the battle. How do you answer? 1, A, B, C, D, like that you answer. Okay, now I see people coming up. So the first question goes, which of the following is an example of a multilocular ovary? Pea, mustard, tomato, wheat. What do you mean by multilocular? As in, basically the ovary is divided into different parts, in different segments. They are known as locules, my dear children. So in which of them do you find multilocular ovary? Answer in 1, A, B, C, D. So it's okay if you get it wrong. Okay, I want everyone to participate. Hi Aisha. Okay, Kavita says A. Okay, Subhashinikannan says C. 1A. Shamna says 1A. Shiv Prasad says C. Let's lock one option. What would be your answer? Which of the following is an example of a multilocular ovary? So I have given a picture. Okay, I have given a picture. Now this would be from your 11th syllabus, my dear children. That is PU1 syllabus, which is you might be confused. So imagine all of this. If you cut them and you see their ovary, how does it look? So all of you, I saw just 1 correct answer, no, 2 correct answer by Srishti as well as Subhashini Kannan. I hope I am pronouncing your name, and also Shiva Prasad, and C saw, yes, the answer is tomato. Let's see, why is it the answer? So if you see the tomato, if you cut it down, you can see something like this. Wherever, my dear children, have you heard about placentation? So basically in the ovary there is placenta, and depending on the position of the placenta as well as the ovules around it, you have different types of placentation. One such placentation is known as exile placentation. Listen to me carefully, you would be able to answer more questions related to this. Okay, this is known as exile placentation, where in the center you have the placenta and around it you have the ovules, right? And basically to divide them you have septa in between. This is known as exile placentation. And where do you see it? You see it in the case of tomato. Apart from that, ma'am, do we have any other options? Yes, you have lemon and also China rose. China rose, lemon and tomato. CTL, all of them are basically examples of exile placentation. Remember that. Okay, so all of you got it correct. Kavita also correct. All of you got it correct. Great, my dear children. All of you got it wrong. It's totally fine. We'll make it correct next time. Okay, Sania, great. Rohan, PDF of all lectures, done, deal today itself. Okay, now which type of placentation is found in pea and bean? Can you guess which type of placentation is found in pea and bean? And I'll tell you where I'm taking these questions from. So you had a experiment, prepare a temporary slide of given material A3 by giving a transverse section of ovary and report the number of locules and the type of placentation. So it's strictly from the practical point of view questions. Okay. Now answer. Which type of placentation is found in P and B? I see the answers. Ramya says C, 2A. Okay, wait. Shiva Prasad says 2A. Mithun says axile. Ramya says C, B. Maheshwari says B. I got the answer. Sania, Nayana, Joseph, hi. Seesaw, Salma, and Subhashini. I will call you Subha. Does it work for you? Seesaw, Reshma, Salma, and Sumitra is confused. Okay. Prakash, good try, bacha. And Trupti, marginal, direct answer. Okay. Molin, oh my God, lots of people have different answers. I'll read out your name. The answer is what? Marginal. Marginal type of placentation is found in P and B. Now, without seeing a picture, you might not understand. Listen, over here, towards one particular margin, you have the placenta. And along with it, you have all the ovules located. Just like this, in P, my dear children, in P, you find it and also in B. So, all of you who got your answers wrong, please rectify it, and all of you got it correct. Kudos to all of you, okay? Now, solve the next question and I'll read out the names of the children who tried to attempt. Now, in which of the following plants is exile placentation observed? With what I explained, you can tell me the answer. With what I explained previously, you can tell me the answer. So, yes, I'll come back to the people who tried to attempt. I had Shivakumar, Shrishti, Supriya, Bhagyashree, and Bhavani, Samartha, and Deepa, Rohan. Pakka, today it will be there. It will be there, okay? I'll ask the team to provide the PDF. Now, Dhriti. Okay, children, can you do me a favor? Whenever you answer, can you say 3 A, B, C, D so that it becomes easy for me to understand what question are you answering? Okay? So, I'll read only the answers who are giving me with the number 3. Hi, Moinli. I am not able to read your name, probably because of that I didn't read out. Yes, you can tell me the name, I'll read it out. Now, 3B. Supriya says 3B, Srushti says 3B, as well as Bhagyashree says 3B. B, yeah. Where do you find exile placentation? Sania says 3C, okay. Children, exile placentation, same like tomato. We saw 3 examples. One was what? China rose. Second one was lemon. And the third one was tomato. China rose is also known as what? Hibiscus. It is known as hibiscus. So your answer would become what? Hibiscus. This is what shows, exile placentation. 3 of them are examples of exile placentation. Remember them. Okay, not get confused. I'll read out all your names, and this is exile placentation. If you do not remember, I put down the picture again. Okay, works? Chalo. Going ahead with the next question. Question number 4. Parietal placentation is present in which plant? Parietal placentation is basically you have towards the wall, placenta is towards the wall, and the ovules would be protruding out like this. So where do you find it? Chili, mustard, hibiscus, and pea. Give your guesses and I'll read out the name for those people who attempted. So there are so many people. I'm so sorry if I'm missing out anyone, okay? So Aisha, Sumitra, Seesaw, Deepa, Vishwa, hi. New people out there. Amulya, Maheshwari, Aarti, Shivakumar, Shubha, and Bhavani, Pooja, Sahab, and then Kiki, Amulya Reddy, Shamna, Aisha. Okay. Now let me read out the answers of the 4th question. So 4th one, let me see who all gave me the correct answer. Parietal placentation is seen in what? Okay, I have B, C, D options coming up. The answer is mustard. Those of you who said B, you are absolutely correct. In the case of mustard only, you will be finding it. So I'll read out a few names. I cannot read out all of them. Samartha is here, and then I have Salma, and then I have Shiva Prasad, Sleepy Sinkhole, oh my God, hi, and Amulya Reddy, Shubha, Seesaw, Supriya, Anusha, Oe, yes, and Srushti. All of you got it correct. Great. So your answer is mustard. With this, we give certain examples, my dear children. Remember, for exile placentation, what is the example? Exile placentation, it is China rose, tomato, as well as lemon. Okay. For parietal placentation, that is towards the periphery, my dear children, you have mustard as an example. Remember it. Okay. And for marginal placentation, you have pea and bean. Now going ahead, free central placentation is a characteristics of free central as in in between you have the placenta, and towards it, basically around it, you would be having what? You would be having the ovule. Now sometimes there could be septum around it like this. Sometimes there could be septum around it. This is known as false septum. This is known as false septum. So basically free central placentation is something that you would be seeing in. What? Sunflower, dianthus, mustard and bean. So my dear children, mustard, you know, it is showing parietal. Bean is showing what? Bean is showing axile placentation. Sorry, marginal placentation. Sunflower is a whole different thing. Now you are left with dianthus. What would be the answer for this? Your answer would be 5B. Exactly. So your answer is dianthus. Remember, so this is the picture of dianthus, okay, and this is how it looks. I have brought the picture so that you can remember it better. So I know it does not look entirely like this, but yes, something around something I hope you are able to understand, okay. Itachi, let's not talk about IPL here, okay, let's about winning KCET, okay, works that. So free central placentation in dianthus, and I'll give you one more example so that you can remember in question if it comes. Dianthus or primrose. Dianthus or primrose. Both of them are example of free central placentation. Do not forget it. Okay, Itachi, I forgive you. No issues at all. Great that you rectified. Now question number 6. Which of the following plants exhibit basal placentation? So basal placentation is like you have the ovary and towards the basal side you would be having the placentation. Okay, you would be having the placentation. Where do you see that? Question number 6. Try to attempt and let me read out the names. It's been long time since I didn't see a lot of my children out there. So I'll just randomly read out names, okay? Deepa, Bhavani, Shushmita, Shubha, Shivakumar, Mallikarjun, hi, Narasimha, Sleepy, Seesaw, Supriya, JJ, a person with my name, Anusha, Ramya, Seesaw, Itachi, okay, Sanya, Moinali, okay, JE student, hi, Mallikarjun, Prakash, okay, Ramya, Jyotika, you scored 98 in bio. Congratulations to you. Super proud of you, okay? Ma'am, once again, 5th question. I'll come back to 5th question. Just give me a second, okay? Let me complete this. I'll come back to it, sub, okay? So, the answer for this question is, which of the one exhibit basal placentation? The answer is marigold and sunflower. Marigold and what? Sunflower. This would be your answer for the question. Okay, so if you see exactly, I didn't get the image, but still I hope you might understand with this particular picture. You might able to picturize. So towards the basal side you have the placentation, which is why the ovule is like located towards the center. Okay, so here from here you have the placenta, that is why it is able to works for you. And children, for basal placentation, example is what? Marigold and sunflower. Now we go back to the 5th question because sub didn't understand it. So free central placentation is basically in the center you have the placenta and around it you would be having the ovule. It's free. It's totally free over here. Example is dianthus and primrose. Works? Great? Cool. Now children, basically we are moving ahead with the next experiment, but before that, I want all of you to write down the answers right now. Axile placentation, 2 example, basal placentation, 2 example, parietal placentation, at least 1 example, okay, or if you can 2 examples, and then what do we have? Marginal placentation and all these examples. Let me see who all get it correct. Okay, and until then, I'll be just reading out the names of the people who are trying to answer the question. Bhavani, ma'am, I got 100 in bio. Oh my god, that's super awesome, Bhavani. I'm very proud of you. Super great. And Sleepy Sinkhole, I scored 100 in bio. Oh my god, even I am very happy. I'm super proud of you. My dear children, Itachi also 89 in bio. Great. And all of you, my dear children, who have not scored great in biology or any other subjects, please do not worry. It's okay. We can give the second attempt. As of now, we have to focus on KCET. And all of you who have scored by learning from us and all, please fill the form that is there because you get a chance to meet us. There's a meet and greet program we are organizing. Please make sure you're doing it. So now let me see who all are putting down the answers. Now, Saqib, it is there in the textbook, ma'am. Yes, it is there, Baba, in the textbook. Okay, JJ says marginal pea and bean. Great. Exile, Deepa says exile is tomato and China rose. Great, awesome. And then Kiki says exile example is hibiscus lemon and marginal example is pea bean. Great. And basal is marigold. So children, Just go through the examples, they are very much important. Basal is marigold and sunflower, remember it, okay. And free central, it is dianthus and primrose, that is free central. And then you have what? Parietal, you have mustard. Parietal, you have mustard. PM, you can remember, Prime Minister, you can remember like that, okay. And then you have exile placentation in hibiscus, tomato and lemon. And also you have marginal placentation in the case of pea and bean. I hope that is done, okay. Now we move ahead to what? To this particular slide. Now, great Kiki and Shubha, basil, marigold and sunflower, great. Parietal mustard, JJ, great, awesome. And Srishti, exile, tomato, China rose, awesome. Trupti, great. And then Samartha, oh my god, he's giving extra examples, great, all very, very good. Arjimon, yes, it's there in your NCRT. Okay, ma'am, please make a proper list of all of them. Okay. Chili. Chili also shows exile placentation, okay. Chili also shows exile placentation. Those of you are asking me an extra thing. Chalo, let's see. So exile, it is for China rose, okay, it is for China rose, and then we have hibiscus, and then we have lemon. And then we came to marginal. Marginal is shown by, tell me, it's pea and bean. Okay, and then what we have free central, that is dianthus and primrose, okay. And then what we have after free central, my dear children, keep on going. I am seeing all of your answer. Then we have basil, yes, basil is what, even I forgot, sunflower and marigold, okay. And then what we have, what am I leaving? Hi Sleepy, Sleepy says I'm Farhana, okay, I'll remember your name. And what are we leaving? Parietal, exactly. Parietal, it is mustard. And my dear Trupti and also one more, Samartha spoke about arjimon. So please remember these examples, very important. Okay, so P for M, Prime Minister. Okay, BSM. There is BMS college, right? BMS college, you can remember like that. BMS, okay. Now free central FD, you have some FD, right? Money you keep in fixed deposit. So F and D, you can remember like that. And marginal is P and B, that you have to remember as such only. And exile is this. Works? So this is all about it. I hope it's clear. Can we go ahead with the next experiment? Give me a thumbs up, give me some hearts, and we will move on to the next experiment. And Aisha, Samartha, Subha, all of you doing really great. Okay? Ma'am, side ho jao. Okay. Take. Okay. Fine. Now we are going ahead. I see some hearts flowing around there. And children whose name I am not able to read, I will be reading it out. Okay, you are doing a wonderful job out there. So prepare a temporary slide to show pollen germination and calculate the percentage of pollen germination, this experiment, and also the pollen tube growth on the stigma and labeled diagram of the observation. This experiment I am going to cover now for the next thing. And all of you who gave hearts and thumbs up, really great. Thank you so much, means a lot. Now question number 7. This should be easy for you guys. This is going to be easy. What are the two cells present in the pollen grain? Tell me, what are the two cells present in the pollen grain? Shubha said 7B, Samartha said 7B, okay. Kiki says B. Pooja, I'll include microbes question next time, okay? Sure. Maheshwari says B, Shamna says B, Deepa says B, Kiki says B. Jai KCET, oh my god. Yamini says B, hi Yamini, Shiva, Pooja, JJ, Prakash, Abhishek, Moin, oh there are so many names. Okay, now I'll come back to the name and I'll answer it. So pollen grain, my dear children, has two types of cells, okay. One would be the big giant cell which is known as generative cell, and the other one will be small cell, sorry, one would be big giant cell which is known as vegetative cell. It became giant, why? Because it was vegetative. Vegetative things do not move around, right? So it became giant like that. And in the cytoplasm of that you have the generative cell, so your answer becomes generative cell and vegetative cell, not endosperm and zygote, not embryocyte and antipode cells, not synergids and polar nuclei. So all of you gave B as an answer. Kudos to all my children, chota champions out there. Really proud of you, okay? I hope all of you got it. Hi Vaishnavi. Now try to find question number 8's answer. Which specimens are commonly used in lab to study pollen germination? Now here you might think rose, vinca, mustard and sunflower, jasmine, marigold. You tell me the answer for this. Jasmine is my name. I hope someone knows that. Okay, 8th answer can you say, and until then I'll read out the name I didn't read. Okay, Moinli and then Vishwa, Kala Vattamma, Vasanta, Honoramma, Muhammad Khizar, Narasimha, Supriya, Shushti, Sumant, Zoe. Okay, Trupti, Sanya, Rashmi, all of you doing great, my dear children. 8th, Itachi, please ask your doubt. Okay, now I see children asking, okay, exactly who said that name? I see someone gave the whole name of the plant. Okay, wait. I somehow lost it. Where is the name? Okay, I saw it somewhere. Okay, anyways, the which specimen is commonly used to study pollen germination? It is vinca, and the exact name is Vinca rosae. Someone said the name. If you could put down again, I would be really happy. The spelling is R-O-S-E-A. Okay, Vinca rosae. If you have not seen it, see it right now. So this is usually taken for pollen germination. If you're asked why, you should know the reason behind that also. Because the pollen viability for this particular plant is really good. It can stay viable for a very long period of time. Kiki, great, my dear child. Okay, great. Itachi, please put down your doubt. I will be answering it. Okay? And all of you who got B as correct, awesome children, going ahead with the next question, that is, what solution is used to nurture pollen grains for pollen tube growth? Which is the solution that we used for nurturing pollen grains for the pollen tube growth? What is going to act like a nutritional medium? Sashi, hello. Okay, Pavan says 9C. Oh my god, really fast. Hi Pavan. Shrishti, Shamna says, and Shubha says 9C. Abhishek says A. Okay, check it out once. Dhruva says C. Aisha says C. Kiki, check. Pavan, Shamna, Sridhar. Awesome. Rashmi, Oe. Roshan. Check, Baba, check once more. Okay. Yamini, Mallikarjun, I'm randomly reading the name. Mallikarjun, please check it once. So all of you who got C as the answer, it is the correct answer. So why do we use sucrose for pollen tube growth? Because sucrose basically does not change the osmolarity. You know, sometimes what will happen, suppose the cells which we are treating with the sucrose solution, they might lose water will be coming out, it will be drying up and things like that. But with sucrose, it does not happen. Sucrose maintains the osmolarity and also basically provides a perfect nourishing medium. It provides energy. Okay, it is a perfect nourishing medium. Glucose is not, salt water is not, even distilled water is not. If you are worried about what is the reason for that, okay, reason is sucrose solution provides necessary osmotic balance and energy for pollen germination. Why not glucose? By glucose is an energy source, sucrose is preferred because it maintains osmolarity. In the case of glucose, it does not maintain osmolarity. There can be problems, water can be moving out and things like that. Salt water, in salt you have so much solute. Anyone, chemistry fans are out over here. So if there is so much solute out there, what will happen? The plant cell will lose water, it will undergo plasmolysis. We do not want that. Distilled water, again, water would be going, there is much more amount of solvent. Okay, so solvent gets inside the cell and the cell bursts. So we do not want all of this. We just want sucrose. Sucrose is the best one. Ma'am, I want to study BSc Nursing. How much rank? Itachi, we will talk about rank for KCET and things like that later. Please put down your things in the comment section. I will be definitely referring to it and I will be answering it. So I hope I am someone who answers to all your, reply back to all your comments. So my dear children, please feel really free to ask that in the comment section. Okay, works? Okay. Okay, next thing. Which chemical is found in the exine of pollen grain? Which chemical is found in the exine of the pollen grain? Moin Ali, we'll come to that question, okay? Tell the answer for this. Pawan says 10B, Sakib says B, Dhruva says B, okay? Shiva says B, Shamna, Samartha, Sridhar, Abhishek great this time, Narsimha, Pavan, Shrishti, Deepa, Aisha, Sumanta, Deepa, all of you doing awesome job, my dear children. The answer would be sporopollenin. In the exine, if you talk about the pollen grain, suppose this is a pollen grain. Pollen grain will have a membrane, plasma membrane. Apart from that, it will be having an intine. One extra layer, it is known as intine. Intine is made up of two things. Remember it always, cellulose and pectin, cellulose and pectin. Now intine feels very lonely, which is why there is one more layer around it which is not continuous, it is discontinuous. There are small pores over here. This discontinuous layer is formed of the most resistant material, that is sporopollenin. Sporopollenin, please remember, very important. So Exine is made up of sporopollenin. Yes, Saqib, and this space which is there, it is known as germ pores. It is known as germ pores. Awesome. Okay, sound the strings, please. Will you guys? Yes, children, yes, we would be doing it. Okay, now going ahead with the next question, 11th question. Try it out. In nature, where is the nutrient medium for pollen germination present in flowers? So in nature, we do not go and add sucrose, right? In nature, the pollen germinates by itself. So what is that thing that provides nutrient medium? Can you tell the answer for that? Anyone out there? Subha says 11C, awesome. Shamna says stigma, awesome. Pavan, A, okay. Check children. Sleepy, great. Sumant, great. Shamna, great. Yashashwini, great. Hi Yashashwini, I see new children out there. Jyotika, Dhruva, Harshita, Jai Shri Krishna. Yashashwini, okay. Mallikarjun, OA, Honorama, Yashashi, Kiki, Moilin Lee, oh my god, Aisha, Sridhar, Muhammad Kizar, awesome children. Sai Deeksha, Ram, oh my god, so many children are telling the correct answer. Yes, the answer is stigma, because stigma is the place where the pollen grain would be coming and landing. So it is the only area where you would be providing nutrient medium. Okay, Jeevan, Aisha, all of you doing really great. Pooja, so this is the answer. I already told you. I hope everyone is understanding as well. Okay, sound the strings. Can you please put that in the comment section below? There are high chances that we could forget it if you put like this. Yeah, child, if you can, please put it down in the comment section. It would be really helpful. Okay, and Moin Ali, you have to wait for that particular answer. I have included that question, do not worry. Now, what does the pollen tube carry? If I say that this is pollen, okay, and basically it has intine and it has the exine. When it lands on the stigma, what happens? There is a tube being formed like this. There's a tube being formed like this. What does this tube carry? That is the question. One male gamete, one egg cell, two male gametes and one tube nucleus, two synergids and one antipodal cell. What is the answer for this? All of you who are saying B, it is the correct answer. Why, my dear children? Listen to me carefully. Those of you did not understand why is it B. Listen to me carefully. So I told you there is a vegetative cell. Agreed? There is a vegetative cell and there is a generative cell. Okay? This vegetative cell will be having a nucleus. Done? Deal? Generative cell, my dear children, what happens to generative cell is that it will be dividing into two cells. And what are these two cells? These are the two male gametes. These are the two male gametes. What? Generative cell divides into two male gametes. So over here, these two male gametes goes in front and it says that, hey, the other nucleus of the vegetative cell, come, you also come. And this is the tube nucleus. This is known as tube nucleus. So tube nucleus is there and also two male gametes, and this becomes your answer. B. Works? Yay! So Kiki is really happy. Chalo. Going ahead to the next question. I want to see who will answer this question. What is the correct formula to calculate percentage of pollen grain germination? Tell the answer and I'll read out the names of those children who answered the question. So I'm just reading out. I do not know if you answered it correct or not. Okay. Sai Diksha, Shrishti, Infinite Kannadiga, hi. And Aisha, Pooja, Santosh, Kiki, Yashashwini, Deepak, Kamate, okay, Muhammad Zoheb, Jyotika, Samartha, Shubha, Abhishek, Sridhar, Yamini, Ramchadda, Moinali, Saqib, Zo, Yashashwini, Deepa, Basana, Oe, Shamna, Jeevan, oh my god, all of you, Vaishnavi, BCP, I'm not able to read out a name, but yes. Okay, so awesome children, you're going great. So those of you who are trying to answer, so I see a lot of you saying A as the answer. Is A the answer or B the answer or C the answer? So Sumitra, Bhuvana, Supriya and Samartha, Krishnamurthy and Harshita says B, I think. Yes, whatever you're thinking is absolutely correct. The answer is this. Okay, the answer is this. N by capital N into 100. What does this N, small n, capital N stand for? What does it stand for, my dear children? Small n is the number of germinated pollen grains. The pollen grains that are going to germinate. N, capital N is what? Total number of pollen grains that we observed. Okay, out of that, only few will be germinating and only few. Since it is few, we will make small n at the top. Okay, small n for few. For the total, we will keep capital N. Into 100 will be your answer. So, your answer is what? B. So, who all got B, my dear children? Awesome. Okay, super good, super good, super good. And children, are you liking the video? Please put the, please click on the like button. It will be really motivating for me when I see it later on. Okay, now try the question number 14. Which stain is used to observe pollen tube growth? Tell me the answer. Safranin, methyl blue, cotton blue, acetocarmine. Which stain is used to observe pollen tube growth? I'm asking about pollen tube growth, my dear children. Okay, Pooja Naik, thank you for the heart. And Deepa. Okay. Deepa, Shushti, okay. Razak, Sleepy, Shamna, Sridhar, Tejaswini. Oh my God, 80 people are there seeing the live. What is the answer for this? What is the answer for this? Hello, Baswaraj, sorry. So, the answer is cotton blue. Cotton blue is something that we use to observe pollen tube growth, my dear children. Okay, none of the other things. Why? What is the reason for that? You have to know the reason, then only you will be able to answer it properly next time. So, cotton blue stains the pollen tube wall because the pollen tube wall is made up of what? Cellulose and pectin. You remember the inner wall, the intine is going and making the pollen tube, right? And it is made up of cellulose and pectin. And this guy, cotton blue, goes and binds on the cellulose and pectin. So, C for cellulose and C for cotton blue, you can remember like that. Now, why not safranin? Safranin stains lignified tissue, wherever there is lignin and things like that. It is there for staining that. Methylene blue, remember this also. It is there for staining the nucleic acids. Okay, nucleic acids, as in you're talking about DNA. Basically, methylene blue is a stain that we use for staining the nucleus. Staining for nucleus. Acetocarmine, it is very specific. It stains the chromosomes. It stains the chromosomes, my dear children. Acetocarmine stains the chromosome. Okay, remember it. So, Kiki says, ma'am, practical questions are also going to be asked in KCET this time. This time it will be asked. Okay, and we never know what questions would be asked, so that's why. So, children, will you remember this? Answer for this pollen tube growth will be cotton blue. Okay, methylene blue is for nucleus staining. Acetocarmine is for chromosome staining. Okay, specifically chromosome staining. And saffron is for lignified. Works? Okay, great. Now, question number 15. Can you try it out? Yes, onion root tip in mitosis, we use acetocarmine. Subha, very good. Okay, now Shamna also putting hearts out there. Ma'am, please upload PDF. Would be definitely doing that. As of now, cannot do it. Okay, it's live going on, but we'll be definitely taking that on priority, Aradhya. Okay. Now, can pollen grains of one plant species germinate on the stigma of another species? Do you think so that can happen? Do you think so that can happen? So, 15 say, I've already got the answer. Akshita says, see, hi Akshita. Pavan and Abhishek and Santosh and Srushti, Sleepy, Yamini. Oh my God. Shashi, Mallikarjun. Okay. Ram Chadda. Awesome, awesome. Jeevita, okay, children. The answer is, can pollen grains of one plant species germinate on the stigma of another species? The answer would be no. Why? Because the pollen-pistil interaction is what? Species specific. So, B is your answer. Okay. Yes, because all plants have the same reproductive structures. Wrong. Okay. Yes, because pollination ensures cross-species fertilization. No, my dear. In that case, probably we would have gone hybrids by ourselves. Why do we do hybridization? Okay. So, this is not happening. No, because all pollen grains are identical and cannot differentiate stigma. No, no, it's not like that. Your only answer becomes what? B. Your only answer becomes B. No, because pollen-pistil interaction is what? Species specific. Okay, children, got it? Will you forget it ever? I hope no one forgets it ever. And going ahead with the next experiment. Ma'am, question didn't understand. Jai Shri Krishna. Which question, Baba? Can you tell me? Number? Jai Shri Krishna, can you tell me the number of the question that you are not able to understand? Just give me a second, children. If someone is not able to understand something, I have to clear it up. Okay. Mohan Lily, I'll tell you one thing. 15th question. Okay, Jai Shri Krishna. Listen to me. Pollen grains of one plant. For example, I have one plant species. Let me take one plant. Rose. Okay. This particular pollen grain, can it go and germinate on a hibiscus plant? Can it go and germinate on a hibiscus plant? No. Why? Because basically the pollen which is going to come and land, over here there is an inspector standing in the stigma region and it checks whether this pollen is of my category or not. So roses pollen grain will be landing on the roses stigma, and if it is totally okay, it will be going ahead with pollen germination. Suppose the stigma is of hibiscus, okay, and this pollen is of what? Is of rose. Whenever hibiscus flower sees rose pollen, it will say that you are not of my category, you cannot basically come and germinate, do the germination. So there is discrimination happening over here. There is discrimination happening over here. So that is why I said that pollen-pistil interaction is species specific. Okay, it cannot be different. Works? I hope, Jai Shri Krishna, now you understood. Okay, now going ahead with the next question. Next experiment that we have. Okay, you're most welcome. And prepare a temporary slide of the given material B and identify any one of the stages of mitosis. So now the questions would be related to mitosis. So all of the people out there who are a big fan of cell biology can answer the questions. Question number 6. Ma'am, please make videos on PCB. How much marks should we get? We will definitely do that, Santosh. Okay, with time we will be definitely doing that. Sure, we will take it on priority. And children, put it down in the comment section all this videos that you want from us, because it will be easy for us to remember then, yeah? Now, I see Narayana saying this is the answer. Which of the following is not a substage of interphase? So, interphase is a very big phase where it is also known as preparatory phase, where preparation of cell cycle happens. Okay, now which of them is not? G1, G2, S phase, telophase. What is the answer for this question? So, I see lots of children answering. Let me read it out first. Okay, let me start. Narayana, Shubha, Pavan, Narasimha, Sridhar, Akshita, Mallikarjun, Shubha, Narayana, Pavan, okay, Padmavati, Umar, Aradhya, great, Jai Shri Krishna, Ram, Tejaswini, Moinli, Samartha, Shushti, great, great, great. The answer for this question is what? D. Okay, one cell divides in 24 hours. So, in that 23 hours it takes for preparation. Okay, for how much time have you prepared for KCET examination? For long, right? But the examination is only for few hours, right? 1 hour each for each subject. Similarly, the preparation is known as interphase. Okay, and basically if you divide that interphase, okay, you have G1 phase, you have S phase and G2 phase. So in G1 phase, some organelles they duplicate. Obviously when there is a cell and the cell has to divide into two, it cannot do that. Take my mitochondria, you take my chloroplast. It cannot do like that. You have to divide equally. So for that, duplication of organelles has to happen. So in G1 phase, duplication of organelles happen, not all of them, very few of them, okay, and the cell grows in size. Remember, whatever I am saying, it's very useful for you. The cell will what? Grow in size, my dear children. Now, in S phase, it is known as synthesis phase, where DNA replication will happen. DNA which is there, it also has to replicate because it also has to be divided, right? So, DNA replication happens over here. In G2 phase, rest of the organelles which didn't duplicate will be duplicating over here. Also, lots of proteins would be made. And finally, you have M phase, where the cell would be actually dividing. Okay. Now, in M phase, my dear children, you have two. One is karyokinesis, where the nucleus divides first, and then you have cytokinesis, where the cell will finally divide into two. Karyokinesis, where the nucleus divides into two, everything is duplicated, now the nucleus divides into two, and then the cell also divides into two. In karyokinesis, okay, there are 4 phases: prophase, metaphase, anaphase, and telophase. PMAT: prophase, metaphase, anaphase, telophase. Okay. This is the thing, and with this theory that I taught you right now, you can answer all the questions coming ahead if the practical based questions come from this particular experiment. Okay, remember interphase G1, S and G2. S is the phase where synthesis of DNA happens, yeah, and M phase is divided into two: karyokinesis, nuclear division, and then cytokinesis, cell division. Okay, in karyokinesis, prophase, metaphase, anaphase, telophase. Interphase takes 23 hours. 1 hour is the time where the cell actually divides. I hope this is completely correct, clear to you guys. Okay, now can we go ahead? So the answer is telophase, because telophase comes under M phase. It is not a part of interphase. Going ahead with the next question. What is the preparatory phase of mitosis? Tell me the answer. Metaphase, interphase, prophase, anaphase. Okay, Anita says D. Anita says D. D for this question. G1 requires 36 ATPs. Okay. Santosh says 17C. Children, preparatory phase. Listen, read the questions carefully. Which is the preparatory phase of mitosis? I told you what is that particular phase in which the cell will take most of its time. It is known as interphase. Remember, over here, interphase. Starting from this particular point, going ahead with this particular point, it is known as interphase. This is where the cell will prepare for division. So your answer should be B. Okay, interphase. So all of you got it correct. Hi Sanjay, big hi dear. Okay, Shrishti, Akshita, I'll read out all your names, my dear children. Go ahead with the next question. Which major event takes place in S phase of interphase? Read it out and I'll read out the name of the children who attempted the question. Santosh, Jai Shri Krishna, Baswaraj, Sridhar, Sanjay, Sanya, Shubha, Nasima, hi, Aradhya, Tejaswini, Muhammad Kizar, Narayana, JJ, Mallikarjun, Varalakshmi, Narasimha, Infinite Kannadiga, and Shrishti, Akshita, Zo, Pavan, Aradhya, JJ, Moinali, Samartha, Anita, Shrishti, Santosh, Sanya, oh my god, Varalakshmi, BCP, Varatma, everyone. Okay? And Samartha, Harshita. Yes, dear, it is all from practical parts. Now, you cannot totally segregate them into practical parts and this. Listen, it can be coming from your, in PU syllabus also, you have all these concepts, right? So, from there also, it could be coming in. So, Moinali Vairusada. Okay, so over here, your answer for this question is, which major event takes place in S phase of interphase? S phase, as I told you, synthesis phase. Synthesis phase. What is being synthesized, my dear children? DNA. So DNA is being replicated. So your answer becomes replication of DNA. Apart from DNA, there is one more thing that is being duplicated. Can anyone tell me the name of that one thing that is duplicated? That is duplicated. Can you tell me, anyone out there? There is one more thing that is getting duplicated during S phase apart from DNA. Array, DNA are the one that forms chromosome. Baba, DNA are the ones that forms chromosome. So when I say chromosome duplication, I mean DNA duplication. No. Cytoplasm. Cytoplasm growth happens in G1 phase. I saw some answer. Mallikarjun, you are almost close. Yes, I think. Centriole. Pavan, yes, centriole. Yes, Mallikarjun and Pavan got it correct. The answer is centriole. Actually, centriole is a part of centrosome. So, centrosome duplication also happens in the S phase. Okay, two things. DNA replication happens, and apart from that, centrosome also replicate. Two things happens. Okay. Centromere is different, dear children. So centromere is a part over here that connects this. This is centromere. That is totally different. Centriole is something that sends spindle fibers. Okay. Centriole is something that produces spindle fibers, and that is a part of centrosome. So we will say, instead of centriole replicating, we will say centrosome replicates or duplicates. Works? Ma'am, I am telling centriole duplicate in the cytoplasm. Okay, then you are totally correct. Kudos, children. Are you understanding? Okay. S phase, what is happening? Two things. Duplication of DNA as well as central duplication. Now, which stain is commonly used to observe the stages of mitosis? Now, I think you can tell me the answer for this. We have discussed this. Which is used to observe the stages of mitosis? In mitosis, my dear children, we are majorly focusing on the chromosomes. Chromosomes are together, chromosomes align in between, they separate and things like that. So, what is that thing that we use for chromosomes? I told you already. 19th, the answer would be 19th, the answer, proper answer. I know you might be confused between two options, but yes, I see a lot of children answering. The answer is acetocarmine. B is the answer. Methylene blue, yes, it basically stains the nucleus, but if you have to be really proper with the answer, acetocarmine is something that will be basically staining the chromosomes as well. So B is your answer for the question. Okay, B is the answer for your question. Ma'am, in S phase, chromosome number increase. It does not. Okay, chromosome number does not increase. Now, going ahead. Next question. What are the two major steps of mitosis? You answer that and I'll read out the names of the children who are attempting. Okay, so I am randomly naming out Mallikarjun, Nakshatra, Zo, JJ, Varalakshmi, Shubha, Bhargavi, and Jai Shri Krishna, Jivita, Narasimha, Srinivas, Srishti, Samartha, Aradhya, Mohammad Kizar, Pavan, and Sanya, Akshita, Aarti, Anita, Sridhar, Manjula Raju, Nayan Kumar, Zo, Bhargavi, Harshita, JJ, Samartha, Shushti, Pavan, Anita, GS World, hi, Vaishnavi, and T is there, Sanjana, Infinite Kannadiga, all of you, Tejaswini. Great, great, my dear children. Tell me the answer for this. What are the two major steps of mitosis? Mitosis, as I said, this is where the cell actually divides. So when the cell divides, the nucleus has to divide, and that is known as karyokinesis. And after the nuclear division, everything is duplicated, nucleus is also divided. You finally divide the cell, which is known as cytokinesis. So here you have B as the correct answer. Okay, Hindu Kesari, okay, cell said let's split, it's time to grow. I said same here, my marks are too low. Oh my god, that's swag. Okay, let's make the mark really, really huge then now. So B is the answer for this question. All of you who got it correct, kudos to you. So my dear children, with this we complete all the practical based questions. Okay, what did we complete? We completed all the practical based questions. Hi Livia. So first of all, we will take 1 minute time. I'll just take 1 minute of your time. All of you understood everything? Did everyone understand everything? I hope you did. If you didn't, what you have to do is, mainly there is a reason I'm not answering, because this question I have included. Okay, that's the reason. So, yes. Everyone understood, everyone is doing great, all are happy, no one is tensed. Even if you are someone who did get your answers correct, it's totally fine. Here you have to study, and this is how we study. We make mistakes, and that's how we study. Okay, so it's all good. Forget everything as in your mistakes. Let's correct all those things and let's learn something new. So that is done, and now I am shifting. Okay, I am shifting my focus to sexual reproduction in flowering plants. I am going to solve questions for certain PYQs, not all of them, certain PYQs from sexual reproduction in flowering plants. Are you ready? Are you ready? Moinali, do not be sad, okay? I am going to answer and read out your name as well when I am going to answer that. Pawan got 94 in both video effort. That's so good. Aarti, will biology be easy? I'll tell you one thing. Biology is actually easy when you study it, like all the other subjects. It's actually, but it does not take a lot of your time to answer. If you know, you know. If you do not know, you do not know. That's all. There is no calculation required and things like that. So yes, it's a scoring point. Make sure you know it. Chalo, let's begin the next chapter. And this question I have marked it in red because here you have to use your brain a little bit. Now, Raghavendra, biology easy. Is biology easy? Then let's make it. Then it's really good. So, a flower has 10 stamens, each having bilobed dithecous anther. How many stamens? 10 stamens are there. Each will have stamen. Each of the stamens will be having anther. Agree? This is the anther, my dear children. So, each stamen, this whole thing is the stamen, and each of them will be having the anther. Fine. Now, if each microsporangium has 5 pollen mother cell, how many pollen grains would be produced by the flower? Samartha, we would be doing a video on that. Okay, dear? So, the question says, listen, taking an anther, if I am zooming it in, it looks like this. Now, do not judge my drawing skills, okay? It might be pathetic on board, but not on in real, but yeah. So, this is an anther. Each of the anther, my dear children, contains 1, 2, 3, 4 microsporangia. How many microsporangia? 4 microsporangia. It is said if each microsporangia has 5 pollen mother cell, 5 pollen mother cell, each of them have it. 1 pollen mother cell, my dear children, how does it divide? It undergoes division, it undergoes meiosis, divides into 2. This is meiosis 1, and then it undergoes again meiosis round 2, and at the end you get 4 cells. How many cells? 4 microspores you get from 1 microspore mother cell. From 1 MMC you get 4 microspores. So over here there are 5 of them. So 5 of them, basically can I say 5 of them will be giving rise to 4, 4, 4. So 5 into 4 is what? 20, 20, 20, 20. Yes, 20 of them. Now listen to me carefully. 20 plus 20 is 40, 40 plus 20 is 60, 60 plus 20 is what? 80. Agreed? You have 80 microspores produced from 1 anther. How many stamens are there? 10 of them. 10 of them will be having 10 anther. So 80 into 10 becomes what? 800. 800 is your answer. D is your answer. Srinivas, and ma'am, your teaching is too good, but I cannot understand molecular base of inheritance. I would be really helping with you that. Okay, I will be helping you with that. Okay, do not worry. There is a video we are gonna make. It will be helpful for you, I hope. Until then, if you can try watching the videos that I have already made. If it does not help, do let me know. So all of you who said 800 as the answer, great. All of you who didn't get the answer correct, it's totally fine. Your answer would be 800. So all of you, I suppose, all of you understood. Give me a heart or give me a thumbs up. Sridhar, Baba, I didn't see what are you saying, please, for. I'm so sorry, I missed it. Can you tell? This question, yes, in the video I have done. So I am including all the PYQs, right? So you might see repetition, okay? Ma'am, please recap you to one shot. Would try so. Okay, next question. Chalo. Thank you for giving me hearts, Srishti and JJ. Means a lot, JS World, everyone. Okay, Moinali, sure. Thank you, Sania, Harshita, everyone out there, Sridhar, Srinivas, Mohammad Kizar, Akshita. Thank you all for acknowledging. Let's go ahead. Nayan Kumar, great attempt, Baba. Which pair of the following cells in the embryo sac are destined to change their ploidy after fertilization? Ploidy as in their number of chromosomes are going to change after fertilization. What do you think is the answer for this? 22A. Okay, so children, my dear children, this is the embryo sac. In the embryo sac, you have 1 and 2 cells. These are known as synergids. These are known as synergids. And there is one more cell which is known as the egg cell. These are what? Synergids. Cool? And over here you have 3 more cells which are known as antipodal cells. And then you have 2 nuclei over here. So this is known as 2 polar nuclei. So this nuclei belongs to a big cell. This nuclei belongs to a big cell which is known as central cell. So when fertilization happens, the pollen grains are going to carry how many gametes? The pollen grains are going to carry 2 gametes. We discussed about that. Generative cell divides into 2, right? So one gamete says that I will come and fuse with egg cell. One gamete says that I'll come and fuse with egg cell. All the cells that you see over here, okay, I'll tell their ploidy also. I'll tell their how many number of, so they are haploid. They are having half the number of chromosome except polar nuclei. If suppose polar nuclei has N, this polar nuclei also has N. If it has 2, then this whole central cell which has these 2 nuclei, what will be the ploidy of the central cell, my dear children? 2N. It is already 2N. Rest all of them are haploid. This is the only one which is what? Diploid. Cool. So one male gamete fuses with this haploid egg. Gamete fuses with haploid egg. Now what do you get? You get 2N. Hasn't the ploidy been changed? Yes, the ploidy has changed. It was N first. Now when it fused, it became 2N. Now what happens? There is one more male gamete. Whom will it fuse with? It will come and fuse with the polar nuclei. It will come and fuse with the polar nuclei. So this particular thing that is coming, my dear children, this particular male gamete that is coming, it is having N. They already have 2N. So it will become what? Triploid. It will become triploid. Again, is the ploidy changing? Yes. What about antipodal cells? They would be like that. What about synergid cells? They would be like that. So who are the ones that change their ploidy? It would be egg cell and the central cell. So all of you who got the answer as A, you are absolutely correct. Kudos to all of you. I'll read out your names. Until then, please go ahead with the next question. One of the 2000-year-old viable sea discovered during the archaeological excavation at King Herod's near Dead Sea was. This was the question asked by Moilli. Okay, and let me read out the names of all of you who have attempted. Subha, BC, Pratma, Shubha, Moinali, and Padmavati, Aradhya, Harshita, Shushti, Sridhar, Malika Arjun, Samartha, Aarti, Deekshita, and Shushti, Jayashri, Krishna, Srinivas, JJ, Sania, Vivek, Mahalakshmi, Mallikarjun, Anita, Mohammad Kizar, GS World, Srinivas, Madivali, Gauri, Ankita. Okay. Abhi, Sridhar. Oh my God, I am tired now. Yes. So all of you. So there was a seed which was viable even after 2000 years, my dear children. So there was a King Herod, okay, and near Dead Sea he was there. what do you call it? The area or kingdom was there. Okay, so there it was found 2000 years old viable seed, my dear children. It was Phoenix dactylifera. It was Phoenix dactylifera. D becomes your answer for this particular question. Now, I'll ask you one question, you tell me the answer. There was another seed which was viable for 10,000 years. This is 2000. One was viable for 10,000 years. What is the answer for this question? 10,000 years. Answer like this, extra. Extra. Answer like this, extra, so that others do not get confused. Okay? Mehboob, Samartha, Shubha, Padmavati, Sridhar, BCP, Chaitra, hi Chaitra, Pratibha. I see so many new people out there. Aradhya, Sania, Mallikarjun, Madivali, and Aarti, Infinite Kannadiga, Harshita. Awesome, my dear children. It is Lupinus arcticus. It was found in the Arctic island. Okay, it was found in the Arctic region. It was Lupinus arcticus. Always remember, this question could also be coming in. 10,000-year-old viability was there. Okay. So, for this question, which was 2000, it was Phoenix dactylifera. Phoenix dactylifera. Going ahead with the next question. Yes, in the Arctic tundra. Great. Awesome, children. Now, 24th question. In the given diagram, identify the parts labeled as A, B, C and D. A, B, C and Baswas Raj followed my opinion. Thank you so much for answering like that. Moinli, I answered your question, dear. Seed discovered during archaeological excavation was Phoenix dactylifera. Okay. Now, yes. 24th, you are trying the answer. My dear children, always remember, I'll explain you something. Yeah. All of you are answering, you're doing great job, my dear children. So, in the case, this is an example of a monocot seed. This is what? A monocot embryo, my dear children. So, what happens is that in monocots, they make one cotyledon. They say that, okay, initially we have endosperm to provide nourishment. In the case of monocot, they say, I'll have endosperm and I'll make some own reserve for myself, and that is known as, what do you call it? That is known as cotyledon. So, they just have one cotyledon. So, it looks something like this. So, they have an embryonal axis. They have an embryonal axis and they have one big cotyledon. And apart from that, they also have endosperm. This cotyledon which is there, it is known as scutellum. It is known as scutellum. Okay. And if you talk about the embryonal axis that you have over here, this particular thing that you see, okay, it is going to be plumule. It is giving rise to the shoot later on. This thing that you have, it is known as radicle, which gives rise to the root later on. The plumule basically, my dear children, it is surrounded by something which is known as coleoptile. Which is known as coleoptile. Radicle, which is giving rise to the root, it is covered by root cap. It is covered by root cap, and apart from that, there is also one more layer out there which is known as coleorhiza. Coleorhiza. Now, the same image, they have put it like this. Okay. So, A part, this is the plumule. So, A part which is there will be what? It will be coleoptile. Okay. So, A is coleoptile. I have two options like that. Let me exclude these options. Now, B. What about B? B is over here. What is B? So, this is the endosperm which is there, the outermost thing. Over here, the white region that you see, extra region, it is the scutellum. So, B becomes scutellum. Again, I am getting this as the answer. Now, going ahead with what do you have? C. What is C? So, C is the covering of the seed in which this particular embryo is there. And what do you call this covering of the seed as? It is known as pericarp. It is known as pericarp. So, C should be pericarp. And what is D? D, here this thing that you see is radicle. Below it, whatever you see would be coleorhiza. Will be what? Coleorhiza. Hence, your answer becomes A. Got it? Your answer becomes A. If you are not able to remember this, you might not be able to remember this as well, which is why I said this is much more easy version for you. Always remember coleorhiza has R in it, so it will be near root. Coleoptile has P in it, so it will be near shoot. Works? Finally, we'll be talking about that answer towards the later phase, so probably I will be commenting it down. Okay? Not covering that particular chapter over here. Do not want to confuse those children who are sitting in this class for another chapter, okay? So, children, your answer would be A. Works? Next question. Question number 25. Consider the following statement and choose the correct answer for the given option. Okay, consider that. So statement 1 says innermost layer of Microsporangium is tapeworm. Statement 2 states that cells of tapeworm possess dense cytoplasm, more than one nucleus, and nourishes the developing embryo. What is the answer for this question? 25B. So Moinali, please Baba. Okay, I'll tell you one thing. If you have questions out of the things that I'm teaching you right now, right away you put it down in the comment section. I'll be helping you there. Why I'm telling you is that there might be children who might not have prepared the chapter and might be coming over here. They will feel very demotivated if I talk about something else. So let's be prepared. Yeah, works? Thank you. If you understand, it will be really great. And thank you for children who are putting down your answers. You're doing an awesome job out there. So if you talk about Microsporangium, so this is suppose the anther. This is the sporogenous tissue where the pollen grains are going to be formed. Okay, outside that, a covering is there which is known as tapeworm. Tapeworm is the one that will be nourishing these cells which are going to form the future pollen grains. Okay, so innermost layer of Microsporangium is tapeworm. Yes, how do you understand? Because this is actually the innermost layer. Outside you have multiple layers. The outermost layer is known as epidermis. It is known as epidermis. Inside the epidermis, you have another layer which is known as endothecium. Okay, epidermis, then endothecium. Now, in between endothecium and the tapeworm, you have something else which is known as middle layer because it is in the middle. So, innermost layer becomes tapeworm, my dear children. That is correct. Cells of the tapeworm possess dense cytoplasm. Obviously, they have to nourish, so it will be dense cytoplasm. Yes, it will be dense cytoplasm, and apart from that, there will be more than one nucleus. Is it true that they have more than one nucleus? Yes, because the cell has not completely divided properly, which is why they have multiple nucleuses. Okay, more than one, basically they have two or something like that, and it is therefore nourishing. So this becomes your answer. So both statement 1 and 2 are correct. B is your answer. Yes, I think it's 2021 question, I'm not sure. In the book it was written like that. Yeah. Now question number 6. In apple, the chromosome number of gametes is 14. What is the chromosome number in its primary endosperm nucleus? Hi Niv. Okay, Nawfal, sorry Baba, I didn't see your question. Can you come again please? Smiley, I'm so good. Thank you for asking that. Madhivali, thank you so much. I'm so sorry I couldn't read out any of your names for so long. So Pratibha, Chaitra, Shubha, Chaitra, Deepa, Bhumika, Chandan, Alan, Infinity, Zoe, Deepa, Tejaswini, Akshita, Sridhar, Moinali, chemistry ma'am is very helpful. Oh my god. Okay, she is absolutely doing a wonderful job. I do agree with you. So what is the chromosome number for this particular question? So in the gamete is being said. In the gamete, my dear children, suppose in the normal cells it is 2n. What will be the number of chromosomes in the gametes? It will be half of it. That is haploid. That is haploid. Okay. So this is diploid and this would be haploid. Agreed? So over here already the gametes number is being mentioned, that is 17. So it is haploid number. We got to understand that. Now what is the chromosome number in its primary endosperm nucleus? What do you mean by primary endosperm nucleus? So I told you basically what happens. The egg cell and the male gamete fuses N and N, you get 2N. And then you had one more thing, that is the two polar nuclei. The two polar nuclei having 2N diploid fuses with another male gamete having N. Right, this is something that will be forming primary endosperm nucleus. This is something that forms primary endosperm nucleus, my dear children. So N and 2N together, N and 2N together, can I say it's 3N? Can I say it's 3N? And what is my N over here? N is 17. So 17 into 3 will be what? 51. It will be 51. Hi Itachi. Only karyokinesis, Baba, mitosis, the whole thing happens inside. Okay, in the, meiosis happens inside. Okay, wait, you're talking about next thing. Only karyokinesis takes place. Yes, exactly. Karyokinesis takes place and cytokinesis does not. which is why they have multiple nucleus out there. Yes? Moinali, please repeat. You didn't understand? Okay, I'll repeat it. Listen to me carefully, okay? In apple, the chromosome number in the gamete, my dear children, gamete will always have haploid, half the number of chromosome than the normal cells, right? So the gamete number is given, the chromosome number in gamete is given, that is 17. Now you are asked about primary endosperm nucleus. So first of all, we should know what is the ploidy of primary endosperm nucleus. So ploidy of primary, how is primary endosperm nucleus formed? When two polar nuclei and the male gamete comes together, that is how it's formed. So two polar nuclei will be having 2n ploidy, okay, and the male gamete has n ploidy. Both of them come together, you have 3n. You have 3n. What is the value of n? N is 17. So 17 into 3 becomes 51. I hope now you understood Moilili. Works? Does it work? I am Spurti. Okay. Copied. Hi Abhi. Ma'am, when was chemistry class? I think it was yesterday. And now I think it will be after a few days. Yes? Harshvardhan. Child, you didn't understand it yet? Harshvardhan. Hello Abhi. Hello Abhi. And okay, chalo. I'll go ahead. Please go ahead. Okay, fine. Among the following statements related to pollens, choose the correct one. Choose the correct one. Statement 1 says, in 40% of angiosperms, pollen grains are shed at 3-cell stage. In 40% of the angiosperms, pollen grains are shed at 3-cell stage. That means basically, if you're talking about pollen grains, they have the vegetative cell. They have what? The vegetative cell, and they have the generative cell, which are already divided into what? 2 gametes. 1 gamete and 2 gametes, which is why this is one cell, this is another cell, this is the third cell that we are having. So do you think in 40% of the angiosperms, they are shed at 3-cell stage? Yes. The answer is yes. Ma'am, I want chemistry class. Chemistry class will be there. I'll tell you, okay, when it's there. I'll put it down in the comment section. So your answer, this is correct. Now, intine is made up of cellulose and pectin. We discussed this. And it is discontinuous with germ pores. Do you think it is discontinuous with germ pores? Intine, pollen grains is surrounded by intine, and intine is not discontinuous. It is a continuous layer. What is discontinuous? It is exine. So until here, the statement was correct. But here, the statement became wrong. So can I say statement 1 is correct, statement 2 is false? Can I say that? Yes. Can I say that? Yes, I can definitely say. So statement 1 is correct and statement 2 is incorrect. C becomes your answer for this question. Okay, intine is continuous. Yes, Trupta is saying. Okay, and let me tell you one more very important thing. If 40% of the angiosperms are giving shedding the cells in 3-cell stage, what about the rest 60%? Rest 60%, my dear children, Pratibha is saying yes, in 2-cell stage. That is, it does not. The vegetative cell remains like that. The generative cell also remains like that. 2-cell, 1 and 2-cell. Okay, 60% goes like that in 2-cell stage and 40% in 3-cell stage. Please remember, it's a very important thing. Previous question, can you just tell me the question number, previous question, by what you mean, as in 26th question or not? Mehfooz Mullah, please ask me the doubt. Okay, children, am I going fast? You can ask me the doubt. Wait, all of you wait for 1 minute. Okay, I know you want to make it fast. There are only few questions left, 35. It's the total questions number is 35. I want that person who was asking me the doubt. Can you tell me what is the doubt in what I taught? Is there something that you didn't understand? What is ploidy levels of antipodal cells? So antipodal, they have N. All of the cells have N except the central cell, because it has 2 polar nuclei. So one has N, one polar nuclei has N, another polar nuclei has N, so it becomes 2N. Okay? Fine. Now, going back to this question, I'm so sorry for others who are waiting. Just give me a second. I'll explain and I'll come back to you guys. Okay? I'm explaining this again. Listen to me carefully. So I'm removing all of this thing. Listen. We have 2 synergids, 1 egg cell. This egg cell which is there, it has N number of chromosome. Okay? Synergids also have N. Antipodal cells which are there, they also have N and N. Okay. Now, there are two polar nuclei over here, and this is a part of a big cell which is known as the central cell. So, if you are asked about the ploidy of central cell, it is N and N, that means it is 2N. Agreed? Now, what happens? You are asked which will be changing, okay, primary endosperm nucleus. What would be the ploidy of that? First, you should be knowing about that. How is it formed? So, one male gamete fuses with the egg cell, okay? So, male gamete has N, egg cell also has N, it becomes 2N. What is this product? This is known as zygote. This is known as zygote. There is another male gamete which comes and fuses with the central cell. Another male gamete comes and fuses with the central cell. This male gamete has N, this guy has 2N already. So, together it will become what? 3N. It will become 3N. And what is this structure? This is the primary endosperm nucleus, which is there for nourishment of the zygote. Agreed? Now, you are said in the question that the chromosome number of the gamete, for example, let's take male gamete, it's 17. So, you know male gamete is always N. The gametes will be always haploid, my dear children. Half the number of chromosome will be there. Okay. Now, what is the ploidy of, or the number of chromosome in the primary endosperm nucleus? So, primary endosperm nucleus is 3N. It is 3N. So, we have to substitute this N value over here. So, 3 into 17, it becomes what? 51. I hope now you got the thing. Okay. Works, Harshvardhan? I suppose now you got the thing. This is the chromosome number in the case of primary endosperm nucleus. If you are asked, what is the chromosome number of a normal cell? The normal cell will not be having half, it will be having 2N. It will be having 2N. So, 17 into 2 becomes something else. Okay? Okay. Chalo. Thank you so much. Thank you for all those you who were waiting. I'm so sorry. Don't tell me anything. Okay. If a tetraploid plant contains 48 chromosomes in the nucleus, then the number of chromosomes in the egg cell and in the synergids are respectively. Now you can tell me, ma'am didn't understand. Naufal, can you tell me what you didn't understand? Children, some people are telling me to go fast, some people are asking me doubt. Do not put me in a situation where I cannot do both. Please try to understand. Yes, I'm giving my maximum over here. Okay. So, if a tetraploid plant cell contains 48 chromosomes, that means a plant is having normal cells in the plant's body is 4N. Okay. So, it is having 48 chromosomes in its nucleus. What is nucleus, my dear children? Nucleus is basically, okay, just a second, lots of people are over here. Yes, it's live. Yes, okay, sorry. So, 4N is equals to 48. Okay, this is a number of chromosomes in the normal cells. The number of chromosomes in the egg and the synergid cell would be what? Egg will always have haploid. That means half of 48 is what? 24. Similarly, synergid also will be having half. All of them had N, right? So, synergid also will be 24. So, the answer would be 24 and 24. Okay? 28, the answer is B. So, all of you got B, you are correct. Now, if you are worried about what is nucleus, so listen, a normal plant cell, okay, a normal plant cell, it has 2N number of chromosomes, agreed. Nucleus is what? In the ovule, my dear children, there are certain cells. There are certain cells surrounding like this. One cell of it is the one that will divide further and give rise to the embryo sac. So, this is just a normal cell, just like any other plant cell out there. So, it will be 2N. So, it will be what? 2N. Okay. And the embryo sac which is there, all of it will be having N and that will be totally different. So, you are asked about 2N. So, I hope you got to understand this. Did you? 28, 28 since, okay, 28B, okay. Works, children? Did you understand it? Yes. Okay. Cool. Let's go ahead with the next question then. In a dithecous anther, yes ma'am, perfect, thank you so much. In a dithecous anther, each pollen sac contains 1000 MMC. What do you mean by MMC? Microspore mother cell. What is the total number of pollen grains produced by the anther? In a dithecous anther, okay, each pollen sac, So suppose this is the anther. Okay, this sporogenous tissue, my dear children, over here, you call this as a pollen sac because it will be, or microsporangium, you call this as a pollen sac because it is the one that gives rise to pollen grains. Now each of them are having 1000 mmC, 1000 mmC as in microspore mother cell. Now one microspore mother cell, my dear children, how many microspores does it produce? So one, when it divides, as I said, it undergoes meiosis 1 giving rise to 2, and then again it undergoes the second round of meiosis giving rise to 4 cells at the end. These are the microspores, and microspores are the one that will be forming what? The pollen grains later on. That will be forming the pollen grains later on, right? So if 1000 of them are there, so I have to multiply into 4. 1000 into 4, 1000 into 4, 1000 into 4, all of them. So I get 4000, 4000 here, 4000 here, 4000 here, 4000 here, right? So 4000 plus 4000 plus 4000 plus 4000. Basically, if I multiply it, I'll get 16,000. So your answer becomes C, 16,000. 16K, exactly. Okay, so this is how you get the answer. Do not be worried when you get questions like this. Fine. Going ahead with the next question, I hope every one of you understood it. Now, yes, which one of the following statement is correct? Read it out and let me read out the name of the children who are out there. I didn't read out for the long time. Okay, Bhumika, Pratibha, JJ, Vivekanand, Infinite Kannadiga, Harish, Pratibha, Moinli, Anita, Shubha, Vivekananda, Abhi, Chaitra, Jeffina, hi, Sania, Sridhar, Madivali, I forgot the name that you said. Yes, but yeah, I know there's a different name. Samarth, Anushri, Infinite Kannadiga, Harshvardhan, Jai Shri Ram, sorry, Jai Shri Krishna, Spurti, Samarth, Amulya, Pavan, GS, Vivekananda, Anushri, all of you doing really great job, my dear children. Let's see the answer for this question. Which one of the following statement is correct? That is the question. Chasmogamous flowers always exhibit gytenogamy. So what do you mean by chasmogamous flowers? The flowers which are open. Which are what? Open. So they have the female and the male part. Yeah. So they, and over here, suppose I am making another flower. Similarly, chasmogamous only, okay? And there is one more flower, that is clistogamous, that will be closed. That will be closed. Now, chasmogamous flowers always exhibit gytenogamy. What do you mean by gytenogamy? Gytenogamy is basically on the same plant, but between two flowers, if pollination is happening, this is known as gytenogamy. Do you think it always exhibits that? No, my dear children. Since it's an open flower, another pollen grains can also come. Another plant's pollen grain can come. This is known as xenogamy. Or probably since they already have pollen grains, so this can also land. This is known as autogamy. So anything can happen. So this is a wrong statement. Okay. Now B. Clistogamous flowers always exhibit autogamy. So clistogamous isn't closed. Do they have an option to take pollen grains from outside? No, they do not have an option. Always it will be showing autogamy. Autogamy is basically when the pollen grains of the same flowers lands on the stigma of the same flower, and it will definitely happen in the case of clisto. CL stands for closed also. So this becomes your answer. Now chasmogamous flowers never exhibits autogamy. Chasmogamous flowers can be exhibiting autogamy, so this is a wrong statement. Clistogamous flowers exhibit both autogamy and gytenogamy. If that happens, then the earth will become something else. Okay, because clistogamous flowers are closed, they do not have any option, so this is not possible. So your answer would be B. All of you got it correct? Okay. All of you got it correct. Let me see all of you who got it correct. Vivekananda, Anita, Bhuvinka, Pavan, Pratibha, Sania, Chaitra, Harshita, Mohammad Kizar, and then GS World, Aarti, Jim Jam. That was my favorite biscuit's name. Okay, Vivekananda, Harshvardhan, Shaila, hi dear. Samarth, clistogamous exhibits self-pollination. Yes, exactly. And autogamy is basically self-pollination. Yes, you all are correct. Infinite Kannadiga, great teacher, your videos are help. Bhumika, you are a great teacher, ma'am, your videos are helping us a lot. Thank you. Thank you so much, and your 100% effort also matters a lot. Ma'am, hibiscus flower beta, who totally 6 members are watching your class live with me. Thank you so much, Moinali. Okay, next question. Shubha is very much serious over here. Before next question, if there is someone who is planning for taking up the series, Abhyas KCET test series, please do that, because we have mock tests put there and it will be really helpful if you have seen, if there is someone who have already taken it. So there's an interface where 60 minutes timer and everything is kept. So you get to an idea of how it will be there on the date of the exam as well. So it is really beneficial for you. The link is given in the description box. You can check it out. And for the friends who have taken Comet K Crash Course, these are the features that are there. Definitely recommended. You can go ahead with that as well. Okay, so works. Chalo. And one very important announcement with that. So we are organizing meet and greet program. Okay, meet and greet program where we will be meeting you guys. But you have to fill the form for that given in your WhatsApp channel. Okay, if you're not joined the WhatsApp channel yet, you should be, and filling the form if you want to meet us. Yes, and it will be there in what you call Bangalore itself. Okay, because we are in Bangalore. Moin Ali, I have solved this question in organism and population. You can check it out. Okay. Next chapter, molecular base of inheritance. Dear, I already did molecular base of inheritance in the previous one. Can you please check it out? And from here starts human reproduction. Okay, from here it starts human reproduction. Hi Free Fire. Okay, let's start. In which month of gestation the first movements of fetus and appearance of hair on head is observed? Can you tell me the answer for this? 31A. Bhumika says A. Mehboob says A, I think. Yes. Shubha A. Chaitra A. And Bhumika A. Anushri A. Samar, Sanya, Shweta, Tejaswini, Anushri, Janilia. Hi. Amulya, Infinite Kannadiga. All of you doing really great. Can I tell the answer for this? The answer is 5th month. So always remember the months. I am here. I'm going to teach you what happens in every month. Listen to me carefully. With that, you would be able to answer all the questions surrounding it. Okay. First month. Loved up. Basically, your heart. When we die, okay, everyone checks our pulse rate and everything like that, because the heart is the major thing that is there. Similarly, when we are born, heart is something that we check. So basically, heartbeat and everything is audible after 1 month. Now, 2nd month, my dear children, the fetus which is there, it will be having limbs like this, okay, limbs and also digits, limbs and digits, and also most of their organ system would be found by then. Most of their organ system and their external genital organ is also formed. So you can do the sex determination over here. Yes. Now, basically, we will skip 3rd and 4th month. Okay, we will skip 3rd and 4th month. We will directly come to the 5th month. Now in the 5th month, baby says that until now, God, you did whatever you want, but I want to show some styling and things like that. So I want hair on my head. So it says that I want hair on my head. God grants it. So there is hair on the head. Hair on the head. And also the baby says I want to move around a bit. So there is movement as well. There is what? Movement. So first movement, first hair on the head is 5th month. Okay, now 6th month. What happens in the 6th month? God says that you did whatever you want. Now listen to me. You should be having eyelids. Your eyelids should be separated. So eyelid separation happens. Eyelid separation happens, my dear children. Apart from that, what happens? Can you guess and tell me? Eyelashes also are formed. Exactly. Eyelashes are formed. One more thing happens. Eyelid separated, eyelashes are formed, and one more thing happens. Anyone out there with the 3rd thing that happens in the end of the 6th month? Eyelid separation, eyelashes formation, and there is one more thing that I leave to you guys. Okay, you have to tell me that. So this is the thing that you have to remember. So when I say first trimester, that means first 3 months. So after the first 3 months, you have heart, limbs and digits and organ system and external genital organs being formed. Then I say 2nd trimester, that means after 6 months. Okay, 2nd trimester as in 3 plus 3, 6. So you have all of this formed in your movement of the body. No, hair on the body, that is the thing. So hair on the head was given in the 5th month because baby asked for it. In the 6th month, God says that you have eyelid separation, you have eyelashes, now you have some hair on your body also. So hair on the body in the 6th month. Okay, fine hairs, exactly, fine hairs, Anushri, Samarth, great my dear children. So will you remember this? Will you remember this? Infinite Kannadiga, great my dear child. So your answer for this question is 5th month. I hope you will remember everything that I taught over here and you would be able to answer. Let's go ahead with the 32 question. Which of the following hormones is not secreted by human placenta? Yes, fully developed fetus by 6th month, but yeah, development still had to take place. 9th month, final development done. Okay. Okay. So which of the following hormones is not secreted by human placenta? Tell me. Placenta is an endocrine tissue. It produces hormones. I hope you know that. There are 4 hormones produced by placenta. There is one that is not. And all of you are watching this video, like the video. If you're liking the teaching, only if you're liking the teaching, otherwise you do not have to. Okay, this is not a forceful thing. Now, yes, so all of you, I see a lot of you are saying it's C and D. So children who are confused between C and D, I'll say D. Okay, relaxin. Placenta does not allow to relax. You can remember like that. Ovary is the one that will be producing relaxin. Okay, placenta produces 4 hormones. Listen to me carefully. Human chorionic gonadotropin, HCG, and human placental lactogen, and then progesterone, and also estrogen. So progesterone and estrogen are already been released by ovary as well, but then later on placenta takes up this job and produces it. So 4 hormones, HCG, HPL, progesterone and estrogen. Relaxin is produced by ovary during later phase of pregnancy. At that point your muscles and everything have to relax, cervical region has to relax so that the baby can easily move out. If it contracts, can the baby move out? No. So that is the thing. Okay, first movement of fetus and appearance of hair on the head, my dear children, 5th month. Okay, chalo. I hope this is clear. Yes, Sridhar is correct, absolutely correct. Ma'am, why not option C? Oh yes, just a second. Okay. That's a typing error. That's a typo error, exactly. Yes. FSH is also not produced. So this is wrong. So I'll say both these options. So I think there is a typing error, which is why it is D and D over here. Okay. Anyways, so FSH is also not an option. You are absolutely correct. If it was estrogen, it would have been better. So suppose let's make it estrogen. Okay, let's make it estrogen. So those of you who have said C as the answer, even though C option was not there, yes, you are also correct. C and D, both C and D. Both C and D works. Thank you so much, Bhumika, for making me see that. Even I didn't spot it out. Okay. Yes, D also. Now, with the thing that I explained you can answer this question. Match the months listed in column 1 with the organogenesis of fetus in column 2. First month what happens, second month what happens, 5th month what happens, 6th month. Amniocentesis. What is amniocentesis? I'll tell you. Amniocentesis, basically the fetus which is there, it is in a fluid. It is covered by a fluid which is known as amniotic fluid. Okay, so there is baby and it is in the amniotic sac, and here you have amniotic fluid. So what happens is that this baby's certain cells would be shutting down, and this particular fluid is taken, and along with that comes the cells. And if you talk about the cell will be having nucleus will be having DNA and everything. So if you examine the DNA and chromosomes and everything, can't you understand the sex chromosomes also? In that way sex determination is done. But actually it was not used for that. It was used for finding any chromosomal abnormalities which is there. Okay, it is for that. So your answer for this question, 33. 33A, ma'am. Okay, let's see. So first month, my dear children, there will be hair on the first month. There will be heart. First month, there is heart. Second month, which is there, what do you have? There is limbs and digits. So A goes with 3. A goes with 3. I have 2 options like that. Let's eliminate other 2 options. Okay. Second month, limbs and digits. Okay. That means B goes with 4. Here B is with 4. 5th month, you have hair on the head. C goes with 2. And 6th month, my dear children, you have separation of eyelids. That is 1. And with that, we get A as the correct answer. So, who all got A as the correct answer? Awesome. Awesome. Okay. And Samarth, it is genetic disorder, okay, not genital. Genital is basically something else. That is in the reproductive aspect. When you talk about that, it's genetic disorders such as sickle cell anemia and hemophilia that you can discover with the help of amniocentesis. Works. So all of you who gave the answer as A, you're absolutely correct. Going ahead with the 34th question. Can you try it out? Identify the cells represented as P this is P. Oh my god, wait. There are two P's over here. Okay, anyways, this is P, this is R, and this is S. So here it is Q. Okay, this is Q. Do not take it as P. 34A, you already gave the answer. So let's see. My dear children, first thing that we have, all of you are correct. Awesome children, it is A. Why A? I'll tell you. So first thing, in the case of oogenesis, you have oogonia. You have what? Oogonia. These cells, they divide, they increase the number and also get differentiated. They get differentiated into something. So P is what? Oogonia. They differentiate into something which is known as primary oocyte. What do you call it? Primary oocyte, and this is your Q. Primary oocyte undergoes meiosis. In meiosis, it happens in two stages. Meiosis 1, where basically it will divide into two. But the twist is, instead of equal division, it will be having a small cell and it will be having a big cell over here. This big cell that is there, it is known as secondary oocyte. This is known as secondary oocyte, and it is R. It is R. And this small thing that is formed is known as first polar body. It is not going to be of any use, so it's gone. Now, again, meiosis 2 has to happen. In meiosis 2, this will again divide into two. Again, there's a small cell and there's a big cell that is being formed. This big cell is known as ovum. And what is S? S is what? Ovum. The small cell is secondary polar body. Again, not of any use. So, which is why if you see, P is oogonia, Q is primary oocyte, and R is secondary oocyte, and S is ovum, which is why A becomes your answer. Okay. Amniocentesis is not a genetic disorder. Moinili, amniocentesis is a process with which you can find genetic disorders. Works? All of you got it correct? Going ahead with the next question. That is 35th. This is the last question. After this, I will be showing you a snip of some picture. You have to tell me the answer. Last question, okay. From the following table, select the option that correctly categorizes various phases of menstrual cycle. Now that is tricky, because why I say it is tricky? Because there is some information that could not match up to what you have studied until now. Okay, works? Chalo. Now let me see who all got the answer correct until now. Okay, I'm just reading out the names of children who have attempted. Jim, Jam, Amulya, Chaitra, Vivekananda, Shaila, Pratibha, Janelia, Samad, no time left. How many years soul enters the... What type of question, Baba? Out of syllabus. No idea. Sayeda, Vivekananda, Bhumika, Crazy Shoot, Anushri, and then Aradhya, Infinite, Samad, Baswara, JJ, JS World, Bhargavi, Pratibha, Moinali, Venkatesh, Mehboob, Spurti, Aradhya, Chandru, hi Chandru, Muhammad Kizar, Sania, Jai Shri Krishna, JS World, Infinite Kannadiga, Sridhar, Jim Jam, Amulya, Saida, Madni, hi Madni, hi Abhi. Okay, is Marigold and Diane? Okay, you come to this different level. Okay, Harshita, I'll answer you, okay? Just give me a second, I'll answer you. Marigold and Dianthus is not the same. No. Okay, it is not the same. Marigold show what type of placentation? It shows basal placentation. Okay, Dianthus is the one that is going to show what do you call it? Free central placentation. Okay, ma'am, it's mine. Fine. Chalo. Chalo. Over here, from the following table, select the options, and over here you are telling me the answer. You are saying D is the answer for this question. Obviously some of the children are saying D. Let me solve it for you. So listen, we have several phases. First phase is M phase, that is menstrual phase. Okay, listen to me carefully if you do not want to attempt mistake on the day of exam. Then is follicular phase. Then is luteal phase. Okay. Over here you have ovulatory phase. This peak portion that you see, it is ovulatory phase. Now, follicular phase is also known as proliferative phase. Proliferative phase. And luteal phase is also known as secretory phase. What phase? Secretory phase. Now, listen to me carefully. During menstrual phase, the endometrium which has formed, listen, fertilization didn't happen. Okay, our body waited for so long, fertilization didn't happen. Then what happened? Basically, the endometrium which was there, it shed down. The endometrium, it shed down. Now, but our body thinks we cannot give up. Okay, body thinks we cannot give up. Basically, we have to reform the endometrium. So, slowly, the endometrium is going to reform. Why? Because next time again an ovum or basically secondary oocyte is going to be released. Our body is going to try once more. So basically over here what happens is that the follicles which were there, it starts regrowing. So in proliferative phase, this is not this phase, okay. In proliferative phase what happens, the follicles are going to get matured. They are going to get matured, and the follicle cells which are there, they are going to release something that is known as estrogen, and this estrogen is going to work on endometrium and make it what? Proliferate back again. So proliferative phase is known as proliferative phase. Why? Because your endometrium is again proliferating, and it is also known as follicular phase because your follicles are also maturing, right? Because of the follicles maturing, you are getting estrogen. This is why this is happening. Now, progesterone, which is the next hormone which is there, it is in stable state. It is not increased as of now. Now what happens? Because of this estrogen being released, okay, because of this estrogen being released, my dear children, what happens is that the particular graphene follicle, the mature graphene follicle, it will be rupturing open and it will be releasing the secondary oocyte which is in there, and this is known as ovulatory phase. This is known as ovulatory phase. So basically when it ruptures, so basically when there is estrogen, this particular estrogen will make another hormone to be released way too much, and that is luteinizing hormone, which causes LH surge, and now when LH surge is there, what will happen? The graphene follicle will rupture and the secondary oocyte will be released. So this is ovulatory phase. This is what? Ovulatory phase. Sometimes follicular phase and ovulatory phase is mixed and given to you. So LH surge basically happens over here. Now LH surge happened, ovulation happened. Okay, now what happens? Whatever is left back, the graphene follicle ruptured and gave the ovum, right? The secondary oocyte. What is left back is known as corpus luteum. Okay, this corpus luteum is going to release a hormone that is progesterone and also another hormone that is estrogen, a little bit more. So what will happen? This particular endometrium will be very, very thick now. Okay, it will be really, really thick now. Children, please restrict yourself to the questions over here which I am teaching. Okay, that will be really, really helpful for me. I will answer all your questions later towards the end of the class. So, corpus luteum released two hormones, progesterone, which is why it became increased, which is why now, since endometrium is very much thickened, it will be secreting certain very important things. Why there is secretion? Because the secondary oocyte is also being released. Suppose fertilization takes place, zygote will be formed. Who will nourish the zygote? This endometrium, which is why secretion also happens, which is why it is known as secretory phase, and which is why it is also known as luteal phase. Why luteal phase? Because corpus luteum is being formed. Okay, works. Now over here, try to find the answer. So in menstrual phase, what will happen? Regeneration of endometrium? No, it is shedding down. Matured follicle? No. Menses is going to happen. Menses means menstrual flow is going to happen. So this and this can happen. This and this can happen. Okay. Now, follicular phase. Developing corpus luteum? No. Corpus luteum develops in what? Luteal phase. LH surge? Exactly not in the follicular phase. Towards the end of the follicular phase, you have LH surge. But let's consider this to be true. Okay. Anyways, this is wrong because it happens in luteal phase. Now going ahead. So you have deleted this option also. You have deleted this options also. That means you're left with just one thing. Regeneration of endometrium happens in the luteal phase. Yes, it regenerates completely. It becomes very, very thick. It will become very, very thick. So can I say D is the answer? Can I say D is the answer? Aradhya, you are asking me to explain once again. I can explain in a second. Okay, listen to me carefully. I'll just sum up everything. In menstrual phase, our body, there is no fertilization. Okay, just taking 1 minute of your time. In menstrual phase, our body realizes there is no fertilization. Endometrium which was formed, it goes down, sheds off. Okay, now our body feels we should try it once more. So slowly, follicles which are there, it will start to mature again. it will start to mature again. And the cells of the follicles, they are going to release something that is known as estrogen. And since there is estrogen, it will tell the endometrium to regrow. So the endometrium slowly regenerates, slowly, okay, in a very slow pace. And follicles are also generating. After one point, because of the estrogen being released, estrogen causes the increase of a hormone that is known as luteinizing hormone. So luteinizing hormone goes way too much. Okay, when the amount of luteinizing hormone goes way too much, what happens? The mature Graafian follicle which is there, it will rupture to give out what? The secondary oocyte which is there. This is known as LH surge, and here ovulation would be happening in the ovulatory phase. Now, the secondary oocyte has been released. What is left behind in the Graafian follicle? It is known as corpus luteum now. It is now known as corpus luteum. And who maintains the corpus luteum? LH only. LH will be maintaining the corpus luteum. This corpus luteum releases two hormones, progesterone as well as estrogen. Progesterone will say that, hey, the secondary oocyte has been released. We have to make the endometrium really thicken. What if fertilization happens? So which is why this thickens up. This would be thickening up, and it will be secreting certain things as well, such that if fertilization happens, it can provide enough amount of nourishment. Works? Works? Ayyo. Okay. FSH hormone. Okay, just a second, okay. Let me answer all of your questions because I didn't see any of it. 34, I will be doing it, okay. I will be doing 34 question once more. Just a second. Ma'am, what's the difference between progesterone and progestrogen? So progestrogen is a synthetic hormone. Okay, is the same thing actually, and progesterone is a natural hormone that you get. Or no. It's basically the same thing. It's basically the same thing. Okay, wait. And what about FSH hormone? So FSH is the one that will be responsible for the maturation of the follicles. It will be the one that will be responsible for maturation of the follicles, and these follicle cells are going to release estrogen. This estrogen which is there, it is going to increase LH, okay, only at one particular point of time. When the amount of estrogen is very much at that particular point of time, it will lead to increase in LH and also FSH. Okay, works? So this is why this is your answer. Shaila, I do not have a class tomorrow, my dear child. Cool. So going ahead with the next thing. You have this. Can you fill in A, B, C and D? This is the last part I have for you. Next, we do not have it. Okay. And until then, you try to solve it and I will be solving a few of your questions. Can you try it out? And children, I hope you're drinking water, you're taking, you're being really safe out of over here. Try solving it. Just a second, okay? I have an important thing to do. Okay. Moinili, morula is a stage of embryonic development. Okay, morula is when the embryo has 8 to 16 cells, it is known as morula. And Sridhar, basically progesterone is the natural hormone and progestrogen is the synthetic hormone. I was correct initially. Okay, progestrogen is the synthetic hormone. Now going ahead. So you are telling me a lot of answers. Let me see all of you who are correct. So this is a scenario of spermatogenesis. This is the scenario of spermatogenesis. Let's see what happens over here. Okay, listen to me. Listen to me. So over here, A is an endocrine secretion which will be giving rise to LH and B. What is this? What is A over here? Any guesses? A will be what? You have to talk about a secretion. It is GnRH, gonadotropin releasing hormone. So who all got A as the correct answer? Who all got A as the correct answer? No one got A as the correct answer. GnRH, gonadotropin releasing hormone. Who releases it, my dear children? It is hypothalamus. It is hypothalamus. Now listen over here. You are not asked who is secreting it. You are asked what is the secretion. There is an endocrine secretion being mentioned over here. You are asked about that. I'll come to you mainly, okay? So GnRH is this thing. GnRH will be going and acting on pituitary gland. Will be going and acting on pituitary gland. Okay? Now this pituitary gland will be releasing two things, that is LH and what is B? So all of you got B as FSH, you are absolutely correct. B will be follicle stimulating hormone. Okay, B will be what? Follicle stimulating hormone. So Mehfooz Mullah, great. Jim Jam got the answer. Okay, I have not checked the entire answer. Okay, just give me a second, I'll tell you. Now, luteinizing hormone will act on Leydig cells. Okay, suppose, listen, in the testis you have seminiferous tubules. In the seminiferous tubules you have Sertoli cells which will be giving nourishment for the formation of sperms. Now, in between the seminiferous tubules you have another cell which is known as Leydig cell which will be releasing what? Androgens. So, LH goes and tells the Leydig cells to release what? Androgens. To release what? Androgens. And androgens, what do they do? What do they do? Tell me that, okay? What do they do? And over here, Sertoli cells are there. What happens to the B cell? So, FSH will be acting on Sertoli cells and Sertoli cells will be releasing certain factors. What are these factors? ABP, androgen binding factors. So, the androgen which is released over here, it has to get inside and it has to be staying at one particular place. For what process to happen? For what process to happen? This process that is there, it is known as spermatogenesis. It is known as spermatogenesis. What is spermatogenesis, my dear children? What is spermatogenesis? Where basically, when you talk about spermatogonia, you have primary spermatocyte, divides into two, you have secondary spermatocyte, divides into two. At the end, you get spermatids. Spermatids are like kids. They are immature, they are round, but we want elongated sperms. So, they undergo a process that is known as spermatogenesis. Spermatogenesis, which basically converts spermatids into mature spermatozoa. For this particular process, you definitely need a lot of androgen, and to make androgen stick to this particular place where everything is happening, Sertoli cells will be releasing androgen binding proteins or the factors, okay, and it will be helping in spermatogenesis. The androgens which are there, okay, the androgens which are there, it is not just working for spermatogenesis, the whole process of spermatogenesis happens over here. So, I will say C is what? Spermatogenesis. C is spermatogenesis. Now, ABP factors are being released so that this particular process at least happens really nicely. Okay. So, what is the answer? This is GnRH released by hypothalamus, yes, and then the GnRH acts on pituitary gland. Pituitary gland is the one that releases LH, luteinizing hormone and follicle stimulating hormone. Luteinizing hormone goes and acts on the Leydig cells, ask it to release what? Androgens. The major androgen, you have testosterone. That is the major androgen. Now this testosterone gets inside the seminiferous tubules and will be helping in the whole process of spermatogenesis, which is why C is spermatogenesis. Spermatogenesis is the whole formation of sperms. Yeah. Now FSH, it will be going and acting on Sertoli cells. It will be going and acting on Sertoli cells. Sertoli cells will be releasing some factors, that is androgen binding proteins, such that the androgen could be coming over here, staying over here and help in the process of spermatogenesis, which basically means conversion of spermatids immature ones into spermatozoa. Works? Works? So this is your thing. Now, those of you who are asking me doubt, you can ask me, and someone asked me primrose and dianthus is the same thing. It's not. Okay, it is not. Hi. Basu Masimat, ma'am, talk about tricks also so that it can help. I'll be talking about certain tricks. I'll be making separate video on that if I can. Okay. Ma'am, why elephant give birth only to baby elephant? What is that question, da? We humans give birth to baby humans only, no? That's why. Okay, children, someone was asking about the, okay. So, first things first, tell me your doubts and then we'll go ahead. Morula and blastocyst. Okay, someone had a doubt. Mainly, since it is of this chapter, I'll explain you. Listen to me carefully. So, male sperm, female egg fuses together, you get the zygote. Agreed? The zygote is single celled. Once the single cell divides into two, it becomes an embryo. Now embryo has different stages. Okay, one is morula where it has 8 to 16 cells, 8 to 16 cells, and each of the cells are known as blastomeres. Each of the cells are known as blastomeres. Okay, and then this morula will again divide. You have blastomeres. There's so many stages. At the end you have one stage where the cells will divide into outer layer trophoblast, and there will be a different layer which is known as inner cell mass. This stage of embryo is known as blastocyst stage, and this is the stage where it will go and bind on the endometrial lining for implantation and will be going ahead. Okay, it comes under cleavage. Yes, my dear children, so zygote, it undergoes cleavage. Cleavage means cell division. Okay, it undergoes cleavage and then later forms morula and also blastocyst. Okay, Bhargavi, when is my next class? I'll tell you. Okay, it will be updated in the group, so please make sure you are in the WhatsApp channel up there. Okay, gastrula also is being formed in between this morula, gastrula, blast, things like that. It goes on like that. In your NCERT, just 2 things are there, so you just have to talk about that. Ma'am, we need last day complete revision. We will be doing that. Okay, so children, I hope I was able. So initially I was thinking that I will be just including practical questions, but I felt that I should be interacting with you live by solving at least few questions. But if you feel that there are a lot of other questions still left, please watch the top 30 videos I have made for separate chapters. Okay, there are 30 questions from each chapter. Please make sure you are watching that. It will be really helpful for you. Okay, was this class helpful? Do you think this class was helpful? If you think, definitely do not forget to like the video and also comment down. And also if you have any doubts related to the videos that we have to make, you can put it down in the comment section. Okay, you can put it down in the session. Ma'am, will related portion come? It would not come. So previous year it came and that was a shocker. I get it, but this time it would not. I am. Okay. Basumi sir, I will definitely do that. And Tarun, okay, already pressed. So thank you, Jai Sri Krishna. So children, I hope all of your okay, lactation explain. So lactation, you know, it's the expelling of the milk, yeah, from the mammary gland. What do you want me to explain that? So the first milk that is being produced, it is known as colostrum, and colostrum is rich in IgA. It is rich in what? IgA. Okay, immunoglobulin A. Remember that as well. Yeah, works. So Aradhya, love you ma'am. I love you guys as well. So children, I think we will end the class. Yeah, because I am also tired. Didn't have my lunch as well. I hope you guys had some amount of food and also you had water breaks. Yeah, works. So ma'am, biotechnology, I have made video on that. Can you please check it? Insemination is basically ejaculation of sperm inside the female reproductive tract. Okay, all of the classes, yes, we would be doing it. Okay, fine. So all of you who were there with me throughout the lecture from the starting point onwards, who tried to answer to all the questions, I truly appreciate your efforts, my dear children. You are doing wonderful and you are going to ace the examination. I'm pretty sure of that. Okay, and lac operon concept. Okay, wait, Mr. Crazy. Okay, he's asking about lac operon. I can explain about lac operon. Listen. Last thing that I'm going to explain, okay? Then we are going to go, okay? Because I am also dead tired now. So, my dear children, in prokaryotes, okay? In prokaryotes, you have operons. As in, for example, this is a gene, suppose. This is the promoter region of a gene, okay? And this is a gene which is known as regulatory gene. Regulatory gene. This regulatory gene can be positive or negative, right? Now, apart from this, there is another gene, and for another gene, you have promoter region, you have an operator region, and there are 3 genes, Z, Y, and you have A. Z, Y, and A. Guys, can you just wait for a second? I have been getting a call for so long. Just a second, okay? I do not know how to mute this, so. Hello? Hello? Are there? Are there? Salam Pudiyadana Pullinjodu eduthu Mandavadi. Pudiyadam Pullinjodu ariyamo Palli ennu vecha Pudiyadam Kappayil ninnu oru 600 meter മുകളിലേക്ക് ഒരു പുതിയ വീട് ഉണ്ടാക്കിയിട്ടുണ്ട്. ജോസങ്ങളുടെ വീടിൻ്റെ ഓപ്പോസിറ്റ്. യെസ്. അല്ല, എനിക്ക് അങ്ങനെ ചോദിച്ചാൽ അറിയത്തില്ല. ചേട്ടാ ഞാൻ ഒരു 10 മിനിറ്റ് കഴിഞ്ഞിട്ട് വിളിച്ചാൽ മതിയോ? ഞാൻ ആക്ച്വലി ഒരു ക്ലാസ് എടുത്തുകൊണ്ടിരിക്കുക. ഒരു 10 മിനിറ്റ് കുഴപ്പമുണ്ടോ? ഇത് പുതിയിടം, പയ്യമ്പള്ളി, പുതിയിടം, മറ്റേ പുളിഞ്ചോട് എന്നൊരു സ്ഥലമുണ്ട് താഴെ. അതെ 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 എക്സാക്ട്ലി. ഓക്കെ, താങ്ക് യു. ഓക്കെ, സോ yes, Kerala 100% literacy. Yes, it was Malayalam. So thank you for waiting for me. I'm so sorry. Yes, now listen to me. Regulatory gene, and this is a separate gene that we have, right? So this regulatory gene is going to produce a protein, regulatory protein, which is negatively going to act. It is known as a repressor protein. It is what? Repressor protein. Okay. Now, what happens is that basically this repressor protein would be going and binding on the operator region. But what is the problem if it goes and binds? Basically, the RNA polymerase which wanted to do transcription for ZYA would not be able to do that because it is stuck over here, and hence transcription would not be happening. Hence these genes would not be expressing their things. For example, Z was responsible for production of beta galactosidase enzyme. Okay, Y was responsible for production of permease enzyme, and A was responsible for production of trans-acetylase enzyme. And why do we need these enzymes? Because in the case of E. coli, this is happening in the E. coli. E. coli is waiting for lactose. It has to break down lactose with the help of beta-galactosidase enzyme to what? To glucose and galactose. So if this enzyme is not produced, this breakdown is not going to happen. It is not going to get enough amount of energy as well. Permease is the one that will, let the lactose enter. It increases the permeability of the membrane of the E. coli. That is the use of permease. And trans-acetylase has other function, as in basically it deals with all the other things that are being produced. So this is the normal thing. So usually in the case, so this is lac operon, and usually it is kept close. It is in switched off mode until and unless lactose makes an entry. When lactose is there, this particular repressor protein, instead of going and binding with the operator region, it will go bind with the lactose. And hence, don't you think now it is free? RNA polymerase can easily move. Yes, RNA polymerase will then move around. And now it is known as switched on condition of lac operon. This is how it works. Okay, works. Children, now I'll be ending up the class. Thank you. Crazy shoot. I hope I was able to explain you. So this lactose is going to act like an inducer. Okay, it is inducing the process of transcription. That is why it is known as inducer. Okay, ma'am, make one-shot videos. We would be definitely making one-shot videos of KCET class. A master stroke is there. Please go watch it. Okay. Function of trans-acetylase is basically breaks down all the things that are formed over here. So it's not a one-stop process. You know, when glucose and galactose is being formed and all the other processes happens, there are so many byproducts that are going to be formed. So it deals with that. Works. So yes, ma'am, exam date. Ma'am, exam date is on 16th you have chemistry and physics, and 17th you have mathematics and biology. And with that I am going ahead. Okay, I would be making strategy videos. All your okay, whatever you want from me, you put it down in the comment section. I'll be making sure that I would try to do it. Okay, so put it down in the comment section, my dear children. Wishing you all the best. Thank you so much for being in the live with me. Bye bye. Take care. Thank you, Zahir. Thank you, Bhumika. And thank you, Sayeda. Every one of you. Take care. Bye-bye. How to join WhatsApp? The link is given in the description box. Okay? The link is given in the description box. Moinali, nalla kutiyai idike. I told in Malayalam. That means be a good boy. Yes, Jayashri, Madivali, Rekha, and everyone. Bye-bye. Take care. Lots of love. Thank you so much. Bhargavi, how to join in WhatsApp channel? Link is in the description box. Please check it out once. Okay, you can join the WhatsApp channel. Bye-bye.